So I wasn't quite able to finish the last few things here I wanted to say about this lab. So here on 17, on the test tube number 2 did change color even without adding succinic acid, and the reason for that is the hamburger meat has all the ingredients that you need. So then the hamburger meat, of course, in the meat is muscle tissue and lots of mitochondria, and the mitochondria, they're running the Krebs cycle all day long, and so all the ingredients that you need in order to run the Krebs cycle, they are present in mitochondria as is. And so buying hamburger meat is a good deal for us because we don't have to buy extra enzyme, we don't have to buy expensive ingredients. Everything you need to run the Krebs cycle is there. And so the answer, that's the answer for 17. Number 18, why did the methylene blue become colorless first at the bottom of the tube yet remain colored on the surface of the hamburger? The reason for that is that when methylene blue is exposed to oxygen, it changes color very quickly, and it turns blue again. So what you see here in these test tubes is that right here on the top of these test tubes, they all stay blue because there's oxygen coming into the test tube, and the oxygen from the air will reoxidize the methylene blue, and so it always stays blue on top. And so the answer, the oxygen in the air reoxidizes the methylene blue, and then the blue color returns. Number 19, why does shaking the test tube restore the blue color? Well, if you shake these test tubes, you're going to mix a lot of air into the hamburger that's here on the bottom, and of course, as soon as the oxygen is reintroduced into the test tube, and then you're shaking and mixing it all in there with the hamburger, then the methylene blue turns blue very quickly, and you see that. So you can restore the blue color by reoxidizing the methylene blue. So that concludes this lab right here, this enzyme lab. I know there was a bunch of stuff here. As far as preparation for the next quiz is concerned, here's what you should do. Go to the beginning of the lab. So here, make sure that you study the important terms, a general enzyme reaction as shown here, and then you need to know all of these reactions that were tested in this lab. So that would include the starch digestion with amylase, the triglycerides, so the dietary fats, the digestion with lipase, or the pancreatic juice we called it, down to glycerol and fatty acids. The protein was digested with pepsin to smaller peptides / amino acids. So you definitely need to know the substrates, which are over here on this side. The products are on this side, and then the enzymes that are generating these products. So you need to know that information for sure. The indicator reaction, please take a look at these indicator reactions. Make sure that what these reactions look like, what it looks like when iodine and starch come together, and simple sugars with Benedicts, and what you have to do and what it looks like. So of course, also then the litmus reagent, make sure that you know what the pH, what it looks like with the pH change. So for each of these reactions, be sure to know what's the substrate, what's the product, and how do we do these reactions, and what we're looking for. So just review the entire lab, of course. Take a look at the images, listen to the recordings, and you should be just fine. One last thing, the next quiz will only test all the hydrolysis reactions, so all the reactions except for the hamburger experiment. I'm going to test you on the hamburger meat experiment together with the next lab topic, which is bioenergetics, and so there it fits better with the Krebs cycle and how muscles burn energy and stuff like that, all of the energetic stuff. So be prepared, and this upcoming quiz, the next quiz, will be on all the hydrolytic enzyme reactions, everything in this lab, in the enzyme lab, except for the hamburger meat experiment, and you will be doing just fine.